Good morning, everybody. This lecture recording corresponds to digestion and absorption. So, like usually, we will start by sharing my PowerPoint presentation. All right, let's have a look at digestion and absorption in animal nutrition. So, and digestion and absorption, of course, is a complex process, but it is formed by successive steps, one followed by another, which actually will go towards the ultimate goal, which is breaking down the macromolecules the animal ingests to simple molecules that are ready and easy and can be easily absorbed, and in some cases with really a low energetic cost. So they're going to be absorbed normally through the epithelium, in the case of monogastrics mostly, and they will actually get into the lymphatic system and from there into the general circulation and reach the different cells and tissues to be used by the animal. So it starts digestion first by ingestion. Of course, the animal will ingest its food, and there will be a first process of mechanical breakdown, normally done by mastication and with the salivation as well, adding and lubrifying the boluses to be ready to be swallowed. So the pieces of food are going to be swallowed normally from the mouth through the esophagus into the stomach or rumen, depending on what type of animal we are talking. We're talking about monogastric or ruminants. So in the case of a monogastric, it's going to be more the emphasis today in this lecture, because we will have a special lecture for ruminant digestion and absorption. So the pieces of food will go through an enzymatic process, but also a chemical process, because if we consider hydrochloric acid like part of a chemical breaking down of cells, but also, of course, it plays a really important role in forming a spatial pH environment, acid environment for the activation of some of the enzymes we're going to see in detail in the next slides. So this enzymatic process that also takes place is mainly done by hydrolysis. So that implies the participation, active participation of water to help break down the chemical bonds, where we talked in our first lecture of this course how energy was actually stored in chemical bonds in food. So this food is going to be broken down then into smaller molecules, mainly to their basic structure unit. For proteins, is going to be amino acids. For lipids, is going to be fatty acids and glycerol and triglycerides. And of course, for carbohydrate will be monosaccharides, and in particular glucose, which is the main fuel, metabolic fuel the cells need to fulfill their role and functions. So nutrients are going to be then go into the circulatory system and reach the different cells in the body. And of course, whatever is not digested by the animal is going to be eliminated or excreted through the rectum, anus, or whatever structure or cloaca the animal actually has. So we can define as a broad concept that digestion is a breakdown of food or food. There is a physical process by mastication and grinding in the mouth by the teeth, a chemical process that some cases can be with or without enzymes, participation of enzymes, and a third process, which is an enzymatical process, of course, where enzymes take an active role. Then it will be followed by absorption, and by absorption we can define it at the actual movement of food particles and simple food particles from the lumen of the digestive tract to the blood or the lymph. So digestion we can define it as the summation of a different process by which macromolecules in the food will be degraded to simple compounds and will be absorbed from the gastrointestinal tract. And we define, and normally in animal nutrition, we use a formula for a concept we call apparent digestibility, which is spread as a percentage. It's a very simple formula, which actually is the nutrients consumed as grams per day minus or subtracting the nutrients that are actually excreted in the feces as gram per day, is divided by the nutrients consumed, multiply by 100, and that will give us a percentage of what we call apparent digestibility, in minus out, divided by whatever was intake, multiplied by 100. And this actually slides show us how the gastrointestinal tract, in particular the intestine, which can be defined, the gastrointestinal tract in general in animals, like a long tube. If we actually manage to dissect perfectly well the digestive tract of any animals, we will end up having a long tube where we might be even able to see through it to the other side, right? So the other opening, with two openings on beginning and end, the mouth and the anus. So of course, as we normally refer in the past in biology, the main mechanisms the organisms have to increase their absorptive area or surface is first by having an elongated organ. Intestine in some animals can reach several meters, and the second mechanism will be by folding itself several times. That, of course, increases the absorption surface of the organ. But then the intestine has a special structure. They actually sometimes are defined or described as finger-like structures, lining the intestine, lining the organ, which are actually called the vilus. And each vilus at the same time, they're actually lined, covered by epithelial cells, and the cells are provided in the apical side of the cell, microvilli, which actually also contributes to increase the absorptive area of the organ. So the presence of a vilus actually multiplies by 1400 the absorptive area compared with the original tube, the intestine considered as a tube. And the presence of the microvilli even takes this increase in absorptive area or surface even higher to an estimated of 25,000 times. So there's a 25,000 time increase in the absorptive area of by the microvilli, the presence of microvilli in the intestinal cells, epithelial cells. And the other thing I want to mention here as well is, and this is not a small detail, is the very rich and intense vasculature that these organs present that runs through the mesentery. So we have a really rich supply of arteries and veins ready to absorb whatever nutrient is going to come through the intestinal epithelial cells, and ready to pick up and take it to the rest of the target organs and tissues that are actually demanding those nutrients to be utilized. And so each villi will be composed by capillaries, veins, and arterial side of capillaries, and also the lymph vessels, sometimes called lacteals. And then the lymph circulatory system actually will play a really fundamental role by draining all these, and especially lipids, draining all these chylomicrons and getting ready to reach the different targets and the different organs in the body. So there are a couple of links there for some short videos which actually explain on a very basic mode the digestion and absorption process of the organisms, especially that's the model used is the human. The second YouTube link is a little bit kind of more for kids, but anyway, but it actually has some really concepts that are pillars for getting a good perspective of digestion and absorption by organisms. So the digestive enzymes are actually all the enzymes that actually participate in digestion and absorption are mainly hydrolytic enzyme, meaning that the chemical bonds of the biomolecules remember to actually free that energy that is actually potentially stored in those chemical bonds to be free. We have the active participation of water. That's why it's a hydrolytic process. So we have, as an example, the case of a sugar, as a generic name, sugar, represented by R, very radical, and then with the presence or the addition of water and the digested enzymes will have a hydrolysis of this sugar into much smaller compound. And this is something that can be also applied as a general model or concept for proteins and fat. So this is the basic mechanism of how enzymes work. So I'm just going to refresh. I'm sure you all have this. I hope you all have this concept from chemistry. So you have the substrate, which in this case could be used, for example, as a model sucrose, which is formed by one molecule of glucose and one molecule of fructose. And the substrate will actually bind with the enzyme, forming an enzyme substrate complex. And here it is important always to stress that normally the active sites of an enzyme is really, really specific. And in some cases, it's so specific, for example, for proteins, that it actually distinguishes between one amino acid and another. So some of the enzymes actually make a big differentiation process or selective process into which amino acid or peptides are going to bind to break down the peptide bond and free the amino acids. But anyway, so we have the enzyme. Again, it's really specific for the substrate, and with the addition of the substrate and the enzyme, we'll start putting a lot of stress on this glucose-fructose bond to break it. And here comes the molecule playing a fundamental role to actually help and break down this bond between glucose and fructose. And of course, the products of the enzymatic process are going to be free, ready to be absorbed as glucose or fructose. Sometimes fructose is actually converted to glucose in the liver in a process called glucogenesis. We'll talk a little bit about that later. And of course, once it is the substrate and the enzyme actually are free, the enzyme will return and reinitiate the same reaction with another molecule of substrate ready to go. So most of the enzymes, I forgot to mention these, are of protein origin. That's another diagram showing this basic action of enzymes on the different substrates. And of course, in the digester process, that can be referred as the destructive process if we consider the breakdown of macromolecules into more simple molecules. We have the addition of a product, the enzyme, forming the enzyme substrate complex with the addition of water, and we'll end up having the formation of the more basic molecules that are ready to be absorbed. And the main role and the main function of enzymes is actually to lower the what we call the activation energy. So the energy that is needed for the chemical reaction of breaking the bonds to go forward and give us our product, which is going to be the more simple molecules. That's what we actually need, and the intestine needs to absorb them. We need, of course, the presence of enzymes, without which we will need much higher energy input from the animal to actually take this chemical process and enzymatic process forward and break that bond, and of course free energy and free the basic molecules we need to use in the different tissues and organs. And one concept that is really important is, remember, enzymes do not actually provide energy to the reaction. The only thing they do is they lower the activation energy for the reaction to take place. But the only molecule that actually does that, provide actually direct energy, is ATP, thanks to that unstable phosphate bond, but that's another story. So this is a very interesting diagram I wanted to include, even though the human is used as a model, where you have what we call the regionalization of the digestive tract, where starting from the mouth to the rectum to the anus, you have all the different secretion and the composition of the different secretions, and how, as we move through the different regions, the pH is going to change, basically going from a physiological or close to physiological pH of 6.5, between 6.5 and 8 to 7. We see how actually the pH really goes down in the stomach, thanks to the secretion of hydrochloric acid, which will be fundamental for the activation of the gastric enzymes. And there you have a summary of most of the most important enzymes we're going to be covering in the next slide. But it's a good diagram that actually summarizes important points. So let's start with water. Water, of course, is not digested, and it's part of a digestive process. And water will actually readily move from the guts to the circulation and vice versa. And the major sites of absorption for water is the omasum. If you remember, is that is one of the 4 stomachs of the ruminant, which actually has the aspect of a book with the different folds that resemble the pages of a book. And of course, the role of omasum is still very clear within the digestive process, but at least we know that it's an important place for water absorption in ruminants. And of course, the ileum and the colon will be the 2 regions of the intestine where most of the water absorption is going to take place in the animal. Now, let's start with the first group of nutrients, which is going to be carbohydrates. We have one component of a carbohydrate, remember, from our previous lecture, specific lecture of carbohydrate was the fiber, of which cellulose, which be the major component of this fiber. And cellulose and hemicellulose, they are actually characterized by the presence of beta linkages between 2 molecules of glucose. And the beta linkages, it actually implies a problem for monogastric because we see that the cellulase, the enzyme that is able to break these beta linkages between molecules of glucose, these actually cellulase and hemicellulase is only produced by microbe, it's not produced by the monogastric or the ruminant. So we actually need the presence of symbiotic bacteria within the gastrointestinal tract to be able to, through the process of fermentation, to break down these cellulose and hemicellulose, which are the major component of the cell plant walls. So we require actually a microbial enzyme to break this bond, and the fermentation process, which actually takes place mostly in rumen. But don't forget that we also, monogastrics also have fermentation process occurring mainly in the cecum and the colon, mainly the cecum. And this is actually an eye diagram that actually explains very well what is the difference between the alpha glucose units and the beta glucose unit. So these are 2 actually interconvertible forms of glucose that are going to differ on the placement of a hydroxyl group attached to the carbon, to the first carbon, sometimes called as carbon 1 or alpha. And for this reason, when the hydroxylic group is on the upside position, is called beta glucose. So the linkage between 2 beta glucose, 1 beta glucose and 1 alpha glucose, actually this what we call the beta 1,4 linkage, which is typical of cellulose, animals are not able to produce that enzyme to break this bond. So that's why we actually need the symbiotic presence of microorganisms in the gastrointestinal tract secreting these cellulase and hemicellulose, being able to break down this beta 1,4 linkage. And in this case of, for example, starch, as a difference with cellulose, all the linkage are actually alpha 1,4 linkages, and that actually does not present a problem for the host, for vertebrates to actually secrete that enzyme and, of course, utilize the starch. But it's not the case of cellulose and hemicellulose. So this is another diagram showing the difference in the linkages between starch and cellulose. Now, the ruminal or large intestinal fermentation that is necessary, as we mentioned, to actually break the cellulose and hemicellulose, the beta 1,4 linkage between units of glucose. We start with cellulose. In this case, sometimes we can have starch getting into the large intestinal, and microbial enzymes will actually break this cellulose and forming glucose units. We can refer as starch, but in the case of starch, starch can also be fermented and utilized in the large intestine. It's not only in the duodenum, as we're going to see, that we can actually break down starch, but it also can be fermented in the large intestine, and the end product is going to be the volatile fatty acid, acetic, propionic, and butyric, that are easily and quickly absorbed through the intestinal walls or the large intestine walls, ready to be used and absorbed and used by the animal. So that's, in this area, we're actually summarizing what the fermentation process actually is. And starches, in the case of starches, they are primarily formed by alpha linkages, and these are the, we can always, to actually identify the presence of these alpha 1,4 linkages, we have to look, a tip I'm giving you is actually look at the hydroxyl group and see what position they are actually down, or they are on the upside down. And this alpha 1,4 linkage can actually, is readily available for both microbial and animal enzymes. So the microbes, and this is important concept here, microbes are also able to break down the alpha easily, no problem, but the beta only by the microbes, not by the animal. So the starches can actually be either fermented or digested. So for the starch digestion, we have a key player here, which we will see is going to be the pancreas. But let's start with the alpha amylase, which is secreted by the pancreas. Sometimes we found alpha amylase in small amounts in saliva, especially in omnivores. But remember, alpha amylase is not present in carnivores and ruminants. So there is no alpha amylase activity on the saliva of the carnivores and ruminants. The other enzymes that are important are maltase and isomaltase, which actually are secreted and produced by the small intestinal brush border. And once the starch and the saccharides, polysaccharides, are actually break down into smaller, simple monosaccharides, they will be, we have what we call sodium-dependent glucose transporters. So that means that they are normally active transporters, so there is an energetic cause for that, and they located in the small intestinal brush border. And the other monosaccharides that can actually be absorbed by the sodium glucose dependent transporter is not only glucose but also galactose, which is one of the monosaccharides forming lactose, glucose, and xylose. The digestibility of starch in general, of carbohydrate, is really high. We are talking about 80% is going to be digested, so it's not really a problem. So once the monosaccharides are absorbed, those that are not glucose are going to be converted in the liver into glucose in a process called hepatic glucogenesis, and these glucose molecules will actually readily use to produce energy by complete oxidation, a process we already saw and we mentioned before many times, or can actually be stored in the liver and the muscle as glycogen to use later. So let's have a look in detail about the digestive process of the saccharides by the animal. We start in the mouth with the secretion of salivary amylase, remember, not present in carnivores and ruminants. And salivary amylase will actually act on amylose, which is one of the most important components of starch. And the amylose will go through the stomach completely unchanged. So we start with polysaccharides. Salivary amylase will break down the polysaccharides into amylose. Amylose will travel through the stomach compartment, and there are no enzymes active for saccharides digestion in the stomach. But then in the pancreas, amylose is going to be converted into maltose and dextrin, and that's by the action of the pancreatic amylase, which is a really important enzyme for saccharide digestion in the animal. And maltose and dextrin is also be converted into monosaccharides by the action of maltose, giving glucose, isomaltose, giving glucose subunits, lactose, which as we know is formed by galactose and glucose, and sucrose is gonna break down into fructose and glucose. So maltase, isomaltase are gonna act on both maltose and isomaltose, producing glucose, as we saw. In the case of lactose, it's gonna be attacked, we can say, or digested by lactase, producing the galactose and glucose we mentioned before. And finally, sucrase acting on sucrose to give us fructose and glucose. This is a diagram actually showing the alpha amylase site of action, which of course is going to be the alpha 1,4 linkage between glucose units. Of course, alpha amylase will not have any activity on a beta 1,4 link between glucose units. We have here maltose and how where's the link where maltase is going to be attacking, an isomaltase on isomaltose to give us 2 units of glucose. And this is a more general diagram that is actually showing us the action, as we saw before, pancreatic alpha amylase acting on amylose and amylopectin. Amylose and amylopectin, remember, are 2 structural components of starch. And alpha amylase from amylose will produce other maltose and maltotriose. And then we have the action of sucrase, maltase, and glucamylase producing glucose units. And the same with the case of sucrose, by the action of sucrase giving us fructose and glucose, and lactose by lactase giving us lactose and glucose. And remember, and this is important, that maltase, isomaltase, lactase, and sucrase, they are produced by the intestine. And finally, the monosaccharides, individual monosaccharides, in this case, in most cases, going to be a glucose, or let's say it's going to be transported by a sodium glucose transporter. 2 molecules of sodium will actually be used to actually internalize that target glucose into the intestinal epithelial cells. And from there, that's from the apical side. On the basal side, glucose is going to get into the blood circulation, and while one potassium channel will be pumping out 1 cation of potassium. And of course, we have the sodium potassium ATPase, which is an active pump, is going to be taking out these sodium molecules that actually were used for the internalization of glucose. But this is an ATPase that's normally present in the cell and keeps that equilibrium of ions within the cell. This is an interesting table because it actually shows the rate of absorption of different monosaccharides in relation to glucose. Glucose considered as 100, so it's a relative absorption or relative to glucose absorption of monosaccharides, and we have in the different species. But for example, in the human, we have that normally, and if you look through the other species like the rat, the human, the pigeon, and the frog, galactose normally has a higher rate of absorption compared, even higher than glucose. And of course, for fructose and mannose, the rate of absorption roughly goes between 30% to 50%. It's not as good as glucose. So glucose is the main, remember, energetic unit used by the cells for their metabolism, and it's the only food the brain, for example, is going to use. So if we consider the large intestinal fermentation of saccharides, we start with the starch. It's going to be converted to glucose by microbial enzymes in the large. We are considering large intestine here, and glucose are actually going to be and produced into volatile fatty acid by the fermentation. So the microbes actually have all the enzymatic battery, the same enzymatic battery as a monogastric has. And in the case of the proteins, of course, they're going to be digesting the peptides, amino acids, and nitrogen, especially in particular ammonia, to produce microbial protein. And glucose, remember, can also be used for the synthesis of microbial proteins by the microbes. And of course, will end up being excreted as microbial protein and eventually being digested, those microbes that pass into the duodenum of the ruminant. So that was the digestion and absorption for saccharide. Now let's get into protein digestion. In protein digestion, we will have two key players, which is hydrochloric acid and pepsin, that they are actually secreted by the stomach. And in the case of ruminants, we are talking, when we say stomach, we're talking about the abomasum, sometimes referred as the true stomach or glandular stomach. And also we will have the presence of other enzymes, which are trypsin, chymotrypsin, elastase, and carboxypeptidases, which are secreted not by the stomach, by the pancreas, into the small intestine by the pancreatic juice. And also we will have the presence of peptidases on the small intestinal brush border. So we have then three compartments that are key for protein digestion, which is the stomach, producing hydrochloric acid and pepsin. When we move towards the duodenum with the secretion of pancreatic juice, we will have trypsin, chymotrypsin, elastase, and carboxypeptidases acting on the duodenum and coming from the pancreas. And finally, in the small intestinal brush border of the epithelial cells that are lining the small intestine, we have the presence of peptidases that are going to be responsible for breaking down oligopeptide, dipeptides into amino acids that are the ones that are going to be absorbed, as well as the peptides. So it is important that we distinguish different enzymes in different compartments. And these peptidases are going to be endopeptidases, aminopeptidases, carboxypeptidases, and dipeptidases. And carboxypeptidases, they owe their name because they actually break the peptide bond between carboxylic groups of peptides, oligopeptides, or dipeptides. That's where the name comes from. So the actually peptide and amino, there's going to be a peptide and amino acid transport, a specific transporter, especially for amino acids, and some of them are highly specific. They actually transport only one type of amino acid. That's also another important concept. And digestion actually depends on the protein quality. So remember, what sometimes we refer as bioavailability is referred to that. It's actually how much of the original protein is going to be absorbed as amino acids at the end of the digestive process. So this is, I don't want you to refer much to the enzyme that I mentioned there, but what I really like, because I will be using another terminology, the ones I really like is of these diagrams, the actual compartmentalization diagram, where we have with a pancreatic phase on the intestinal lumen, where proteins are going to be breaking to peptides. Some of them are going to give us amino acids, which are going to be readily absorbed into the intestinal cells and ready to go into the portal vein. And some of these proteins are going to form peptides. We're going to be break down by exopeptidases into small peptide. So the amino acids that are formed again in the brush border oligopeptidases are going to be transform these small peptides into amino acids. Again, going to be absorbed through the brush border membrane of the intestinal cells, internalized into the cytoplasm, and go through the portal vein into the circulatory system. And then the small peptides, again, is going to be break down into amino acid or small peptides. Some of them, small peptide, dipeptides, can sometimes be absorbed directly, and they will go again into the portal vein, into the circulatory process. Now, let's have a more detailed look at the different enzymes that actually work on the digestion of protein. So we start with the mouth. No peptidase is present in the mouth. And we move directly into the stomach, where we have the presence of the oxyntic cells in the fundic region of the stomach, secreting hydrochloric acid, which is going to actually produce a pH environment, an acidic, really acidic environment between 1.5 to 2.5, to give you a reference, which is really acid. And that will actually contribute to the transformation of pepsinogen into pepsin. Pepsinogen is a proenzyme secreted by the Chief cells. And so pepsin is going to be fundamental for the breakdown of proteins, and also we will find in the stomach another enzyme that is normally present in high quantities in young lactating animal, which is renin, and it is the enzyme responsible for attacking the milk protein casein and forming the curd of the young lactating animals. And then we move, we will have actually enterokinase, which is going to be responsible, present in the intestinal cells, which is going to be responsible for the transformation of trypsinogen into trypsin, secreted these enzymes by the pancreas. And this will actually produce peptides, smaller peptides, ending in basic amino acid, arginine, histine, and lysine. And then we have the presence of chymotrypsin that is going to be converted from chymotrypsinogen, also secreted by the pancreas, and will form peptides ending in aromatic amino acid, phenylalanine, tryptophan, and tyrosine. And one thing that is important that I forgot to mention is when trypsinogen is transformed into trypsin, the activated trypsin will also contribute kind of positive feedback in itself to produce more activated trypsin. Anyway, so chymotrypsin, trypsin, forming peptides ending in different types of amino acids. This actually, I'm telling you this because I want you to show and get the concept of how specific these enzymes can be. And then trypsin will also be the one responsible for activating chymotrypsinogen into chymotrypsin. And then we have elastase, which is secreted as proelastase, and this proelastase is going to be activated to produce active elastase by, again, trypsin. And in this case, elastase will form peptides that end in aliphatic amino acid, alanine, glycine, leucine, and isoleucine. And then we have carboxypeptidases A and B, which are secreted as procarboxypeptidases. And if you look carefully here, you see that carboxypeptidases A actually attack those peptides that end in aromatic and aliphatic amino acids that actually were the ones formed by chymotrypsin and elastase. And carboxypeptidase B actually will attack peptides that end in basic amino acids that, if you see from above, were those peptides formed by trypsin. And again, trypsin is responsible for activating both carboxypeptidase A and B from procarboxypeptidase in the pancreas. Again, this is a enzymatic process that takes place in the duodenum and is thanks to the action of a pancreatic juice containing these really crucial enzymes for protein digestion: trypsin, chymotrypsin, elastase, and carboxypeptidases A and B. And finally, those peptides, D-peptides that were produced, or oligopeptides, are going to be attacked by the intestinal cells that actually secrete aminopeptidases. So they're going to form D-peptides, and also these D-peptides at the same time are going to be digested by D-peptidases enzyme producing amino acids, and then they are ready to be absorbed. Okay, and the other thing, if you see this diagram as a whole, you will notice how one enzyme is responsible for breaking down the product of a previous enzyme, and so on. This is a continuous process as in this case, the protein moves along the gastrointestinal tract from the mouth to the intestine. And finally, this is a good diagram that actually shows the internalization of the amino acid by amino acid transported proteins. And sometimes peptides, dipeptides and tripeptides can actually be also internalized by other transport protein, and that activity actually takes place on the cell membrane of the enterocytes. And this is a diagram I like very much because it's very simple, but it shows, but it's very complete, and it shows you how we start with the protein and the action of pepsin and pancreatic proteases actually forming small peptides which have their own carriers, and they're going to be internalized into the intestinal cells. And there are some intracellular peptidases that are going to be in charge of breaking down those small peptides into the basic amino acids. And of course, they're going to be ready to be absorbed from the basal side of the intestinal cell into the capillaries. In the case of more large peptides, of course, we will need the action of the peptidases present in the brush border of the intestinal cells, breaking these more large peptide into smaller peptides that can actually either be internalized or the production of free amino acids. And here, as we see, we have distinguished neutral, basic, and acidic amino acids that have specific carrier-mediated transport, and they're going to be internalized and also be absorbed into capillary circulation. So we have, again, in the intestinal cells, peptide transporters and specific amino acid transporters. Now, let's get into lipid digestion. In this case, it's a more complex digestive process because it implies the breakdown of triglycerides and the re-esterification of triglycerides from the basic monoglycerides and free fatty acids in the endoplasmic reticulum within the cell. But let's have a look at the lipid digestion. So first, we have the fundamental role of bile. The bile is necessary for the emulsification of lipids. Remember, lipids are not soluble in water, so we actually need to form smaller droplets of these lipids so the enzymes are able to attack them and digest them. So the main role of bile will be bile salts will be to emulsify lipids and form the smaller lipid droplets. And then we have the action on pancreatic colipase and lipase, which is going to be responsible for the digestion of lipids. But colipase, in this case, actually has a role of displacing the bile acids, so the enzyme lipase can actually attack the lipids. And lipase will actually cleave the fatty acids and form monoglycerides and two free fatty acids from triglycerides. And we have the absorption of the fatty acids is going to be much easier because of their lipid nature of the nutrient we're considering. So they can be easily absorbed by diffusion by the cells. Remember, membranes, cell membranes are formed by phospholipids, so that doesn't actually imply much of a barrier. And fat is also usually highly digested. We're talking about more than 80%. And we have these, so we start with the clam fat being ingested and the pore of bile, which are gonna separate and form micelles in a process called emulsification. And that will allow the enzyme to actually attack these fats, producing free fatty acid, which is what we will actually, the cell will absorb and utilize in the different target tissues for their requirement. So we start with the emulsification process. We can see this diagram where these micelles are actually associated with molecules of cholesterol, of course, the triacylglycerol, phospholipids, and the bile salts. And these we will have that the action of lipases, phospholipases, and colipases are gonna act on these micelles, producing diacylglycerol fatty acids and also lipophosphatidic acid and monoacylglycerols and forming these mixed micelles. So the mixed micelle is formed by bile salts on the periphery, and also the presence of cholesterol molecules and phospholipids and the lipophosphatidic acid. So this mixed micelles, which is a kind of a mixed structure, that's why it's called mixed micelles, will actually get in contact with the brush border of the intestinal cells, and lipases were going to act, and we'll have the absorption of free fatty acids and cholesterol, and there will be again a re-esterification process inside the intestinal cells, and we will have the formation again of a re-esterification process of triacylglycerides, which are going to be and then associated with apoproteins, which are going to be in charge of absorbing from the from the basal side of the intestinal cell, the new chylomicrons, which are going to get into the lymphatic circulation, and these will going to reach the general circulation and through the lymphatic system, and actually reach the target cell in the different tissues and cells that actually require, and can of course are going to be either deposited as adipose tissue or used for energy production. So let's have a look at this process more simplifying this diagram. So we start with the bile cells responsible for actually producing this emulsification process of dietary fats. So forming these mixed micelles, remember, formed by phospholipid, lipophosphatidic acid, and cholesterol. And these intestinal lipases are going to degrade these triacylglycerols, producing free fatty acids and monoglycerides. And these monoglycerides are going to be associated with apoproteins, which are going to be triacylglycerols, are going to be incorporated with cholesterol and forming the chylomicrons. And these chylomicrons is the way the lipids are going to be actually transported or circulate in blood. And these chylomicrons will actually move through and get into the lymphatic system, reaching the bloodstream. And in the capillaries, remember, in the endothelium that is actually lining the capillaries, we will have lipoprotein lipases that are going to release in the capillaries the fatty acids and the glycerol, which is going to be internalized into the adipocytes and form either are going to be used for oxidation, in an oxidation process to produce energy, like could be the muscle, or are going to be re-esterified again to form storage lipids, and of course it's going to be forming more adipose tissue. Now, to finish with minerals and vitamins, well, there is a huge variation on the digestion or absorption of minerals and vitamins. Of course, liposoluble or fat-soluble vitamins normally are going to be absorbed with the lipids in the same digestion process, because they are dissolved in the fat. But sometimes it depends on the mineral vitamin, may or may not be broken down before digestion. Most vitamins are absorbed as they are, but in the case of the minerals, it's highly variable. Each mineral has its own mechanism of absorption. I'm going to show you two as an example. So most of the absorption of minerals and vitamins occurs again in the small intestine, and there are many absorption systems depending on the individual mineral or vitamin considered. So it's going to be quite variable, the digestion and the absorption. But in the case of minerals, if you remember, the absorption will depend a lot on the mineral source we are considering, if it was an inorganic source of minerals, or we were actually considering the chelated minerals, which the association with amino acid and oligopeptides actually increase the absorption and the digestion absorption of minerals by more, if I don't recall wrong, 600% higher than the inorganic sources of mineral. Of course, they were more expensive, but they actually presented a much higher absorption and metabolism. So this is an example of one mechanism of the regulation and absorption of copper. So you see it's very, very specific, and they go through different transformations, reduction process, sometimes or oxidation process within the intestinal cell to be absorbed by the enterocytes. This is, for example, the case of the zinc transport pathway. Again, it's a very specific process, and it will depend on the mineral we consider. Of course, if you're interested in one mineral in particular about the absorptive process, please let me know and I can provide that material for sure. So as a kind of review of this lecture of absorption of digestion, with an emphasis, of course, consider on the monogastric, we will have a specific lecture for ruminants. We started with the starch, where pancreatic and small intestine enzymes are key. There is glucose transporters that exist in the small intestine, the main place of absorption of glucose. Then we went through the protein absorption and digestion, where pepsin and hydrochloric acid were fundamental in the stomach, but of course the consecutive action of pancreatic and small intestinal enzymes to produce peptides and amino acids, which were transported by specific transporters, sodium-dependent, sometimes transporters. And we have the fat with the action of the bile salts, which are fundamental for the emulsification process, so allowing the enzyme to digest lipids, and the intervention of pancreatic enzymes, where are lipases, which are fundamental for the breakdown of triacylglycerol, producing monoglycerides and free fatty acids, and the formation of the chylomicrons, which is the way lipid circulates in blood, and re-esterification also processed then within the cells. And then we have the absorption mainly by diffusion, no transporter for lipids. That's due to the lipid nature of these nutrients, which actually are easily absorbed by the cell by the phospholipid membranes in the cells. In the case of a large intestine, we have a completely different scenario with a lot of microbial activity in a symbiotic way. And we have the fermentation process of some carbohydrate like starch and fiber. And remember, the microbes were able to break down the beta linkage, 1,4 linkage, that pass through the small intestine, producing volatile fatty acids, which are going to be absorbed. Remember, the glucose homeostasis in the ruminant is not as the one in monogastric. Glucose is mainly formed by gluconeogenesis from the volatile fatty acids in the case of a ruminant. So they also, in the large intestine, there is a conversion, some of a protein that passes through the small intestine to the large intestine by the microbe, but most of it is going to be excreted. And the large intestine may be able to modify lipids that pass through the small intestine from the small intestine, but there's not much absorption of lipids in the large intestine. So most of the absorption of lipids, remember, occurs in the duodenum mainly. But one of the main roles of a large intestine, of course, is going to be water reabsorption. So with that last slide, we finish this lecture. Thank you very much for your attention.